Assalamu alaikum y'all. Welcome back to my YouTube channel. I am back with another educational video on my favorite subject, biology. So let's start with the chapter, the cell. In these video lectures of mine, I will be solely targeting the entrance exam or the aptitude test. So we'll cover up all the pointers that one must know about the topic cell before appearing for an entrance exam. So yeah, let's get started. Okay, so in these lectures of mine, I'm going to keep things very precise and I'm going to share with you only the key pointers that you must know and from where the MCQs are made. So yeah, to begin with, most importantly, we must know the definition of the cell. So the cell can be defined as the structural and functional unit of living organisms and is the smallest unit that can carry out all the activities of life. So the key words are structural and functional unit of life and the smallest unit of life. So yeah. Secondly, the cell was discovered in the 17th century. This is clearly an MCQ. So yeah, note that. The term cell was first used by Robert Hooke in 1665 to describe his investigations on the structure of cork, and the term was first used in the book called Micrographia. Inside the cell, a spherical body called the nucleus was discovered by Robert Brown in 1831 in the cells of orchids. Later, in 1839, the scientist named Shawn described the cell as a structure which consists of a nucleus surrounded by a semi-fluid substance enclosed by a membrane. This definition is important, and you must know that this specific definition was given by Shawn in 1839. Next, a newborn human baby contains 2 trillion cells and an adult contains 60 trillion cells. When you donate blood, you give away 5.4 billion cells. Each day, a human body sloughs off and replaces 1% of its cells, or about 600 billion cells, by the process of mitosis. Now let's talk about the emergence of the cell theory. After the discovery of cell, a lot of information has been collected by different researchers. This information has been summarized in the form of cell theory. The cell theory was formulated by Schleiden in 1838, Schwann in 1839, and Rudolf Virchow in 1855. There are 3 general postulates of the cell theory. Firstly, all organisms are composed of one or more cells. Secondly, the cell is the structural and functional unit of life. And thirdly, cells can arise only by division of pre-existing cells. It is not a de novo structure. The third postulate was given by Rudolf Virchow. Now let's quickly discuss about the microscopes. The compound microscope was invented by David Janssen in 1590. Later in 1610, Galileo designed it properly. You must also note that the resolution power of compound microscope is 250 nanometer. And you must also note that the resolution power of naked eye is 0.1 millimeter. Now you may note the definition of microspory. So the definition is that it is the science of investigating small objects using microscopes. There are various kinds of microscopes depending on the source of light. For example, the light microscope, in which visible light is used as the source of illumination. Then we have X-ray microscope, in which short wavelength X-rays are used as the source of illumination. And finally, we have electron microscope, in which electron beam is used as the source of illumination. Now there's this comparison between the light microscope and the electron microscope that you must be aware of. To begin with, the source of illumination for light microscope is simply the visible light, and for the electron microscope is the electron beam. The magnifying power of the light microscope is 500 times of the naked eye, and that of the electron microscope is 250,000 times of the naked eye. The resolving power of the light microscope is 2 micrometer, and for the electron microscope, it varies between 2 and 4 angstrom. A glass lens is used in the light microscope, whereas an electromagnetic lens is used in the electron microscope. The light microscope is normally used to observe objects that are larger than 200 nanometer in size, and the electron microscope is used to observe objects that are smaller than 200 nanometer in size. These pointers of comparison are very important and have been frequently asked in the MCQs. Sometimes they also ask you to calculate the magnification of a microscope. So it is calculated by multiplying the magnifying power of the eyepiece with the magnifying power of the objective lens. Now let's discuss about the different techniques that are used to isolate the components of a cell. So let us begin with cell fractionation. Cell fractionation is the combination of various methods used to separate the cell organelles and other components based upon the size, and density of the medium. The principle of cell fractionation consists of two steps: homogenization and centrifugation. Homogenization involves the grinding of the cells in a suitable medium with correct pH, ionic composition, and temperature by using a blender. In plants, pectinase is added to digest the middle lamella. The procedure gives rise to a uniform mixture of cells which is then centrifuged. Centrifugation is the process to separate substances on the basis of their size and densities under the influence of centrifugal force by using centrifuge. In density gradient centrifugation, the cell components of different sizes and densities are separated in different layers. The upper layers are less denser than the lower layers. In differential centrifugation, the size and shape of a particle determines how fast it settles. At each step, the content which settles in the bottom of the tube is called the pellet, and those that remain suspended above in the form of liquid are called supernatants. After each speed, the supernatant can be thrown off and centrifuged again. A series of pellet containing cell organelles of smaller and smaller size can therefore be obtained. Lastly, the basic components of a cell include the plasma membrane, the cytoplasm containing the cell organelles, and the nucleus with nuclear or chromatin material. A cell wall is also present in plant cells. This was it for today's lecture. This is just the introduction to the cell biology. There is much more to come. The video had crossed over 10 minutes, so I had to speed up my recording because I understand that as medical students, your time is very precious. These are all the introductory pointers that must be on your fingertips, and I have extracted these pointers from the books of the four provinces. So, yeah, they are completely reliable, and they are the ones that I used to prepare for my entrance exam.